I work in the physics department in Edinburgh, in particular the soft matter physics department. And a lot of you might ask, why are physicists interested in wastewater treatment? Well, hopefully I'll give you a little insight into why that is. Firstly, I'd like to thank the organizers for setting up this event. It's really cool, and it's good to see different people doing different types of research all over the world. So in soft matter physics, what we are interested in is trying to develop simple model systems to understand complex viscous media, essentially. So think paint, think chocolate, but also think about aggregated bacteria systems such as biofilms, where you have cells secreting extracellular polymeric materials. And so these are soft and squishy. Okay. And what my research is mainly concerned with is doing experiments and simulations of aggregated bacteria that assemble on surfaces and in suspension. So on the top row here, you'll see an experimental system of aggregated bacteria. You'll see a governing equation that describes the fundamental physics of this system, and then below a corresponding computer simulation model that helps us describe the system above. So where's the link to wastewater treatment? So here is a bird's eye view of the wastewater treatment facility in Edinburgh. It's an activated sludge plant. And here we see the aeration tanks, and we've heard a lot about that already today. If we were to zoom in one of these aeration tanks, okay, and if we were to take a sample from the aeration tank and visualize it under the microscope, we would see a microbial aggregate here, a bacterial aggregate, or FLOC, as they're referred to in the wastewater treatment industry. And so essentially, this is like a floating biofilm. So you have different, I like to represent it schematically because I'm a physicist and I like to think about things simply. So to me, this is a system of filamentous cells, rod-shaped cells, maybe some spherical cells, and they're all stuck together by some sticky matrix. So this overlap with the soft matter physics and aggregating bacteria and wastewater led me to have meetings with Paul Banfield, who spoke earlier at Veolia. And as a result of the interactions that we've had together, we've embarked on some collaborative research. And that research is now focused on three main strands. One where we look at the role of filaments in aggregation, and I'll talk maybe one slide on this very, very briefly. And then also another brief slide on model systems for N2O, but mainly today I'll talk about machine learning for sludge characterization. Okay, this is these arrows represent time. So this small arrow here is representative of the fact that we haven't actually done any research on this. We've just applied for a PhD. So firstly, we are interested in trying to develop model systems to understand filamentous aggregation. As Paul said, filaments can be problematic in activated sludge. And what we do is we use this organism called Commomonas denitrificans that has been isolated from activated sludge. And it has a propensity to filament, as demonstrated here, and by these cell length distributions along the bottom. And it also can aggregate together. So we've won our proof of concept award from the National Biofilms Innovation Centre along with Veolia to try and develop this organism as a model system. We've also just proposed a PhD project to try and use Commomonas denitrificans. The name denitrificans implies that it is involved in denitrification processes. So we're trying to use this as a model system to understand N2O production and consumption in N2O in wastewater treatment. And as we know, N2O is a greenhouse gas, but what a lot of people don't know is that it's 300 times more potent than carbon dioxide in the atmosphere, but we just produce a lot less of it in society. So now on to the main topic of my talk. So it's the use of machine learning in wastewater treatment. Now I should say from the outset, I am not an expert in machine learning. We just started using machine learning as a tool to try and help us understand processes in wastewater treatment. So if we go back to our aeration tank, on a good day, we may take a sample, visualize it under the microscope, and we would see something like this. This is a good, dense, compact floc with good settling properties. Okay, but on another day, depending on the conditions, etc., we may see something like this. And here we see that this looks rather different to that. We don't have these dense, compact flocks anymore. We have a rather diffuse structure. And this is because these filaments now have become too abundant and are taking over the system and preventing compaction and hindering the settling. So why do we care about this? Well, flock structure can give rise, or unhealthy flock structure can give rise to unhealthy sludge, which then gives rise to settling problems in the activated sludge process. And this is sometimes unpredictable. And the state of the art at the minute is basically operators visualize the samples under the microscope and then ascertain whether it is healthy or unhealthy flock or sludge. And so it relies on the experience and intuition of the operator. But they also complement these microscopy images with an indicator for sludge health called the sludge volume index, which Paul alluded to. And basically this measures the settability of the sludge. And it ranges in values between the maybe 10, 20 to 300. And so what I'll show below now is here are representative images of sludge samples that belong to these three categories. So here we have a sample that is too dense. Okay, so that it settles too quickly and doesn't really flocculate as it settles. And then this is a sample that is too fluffy. So it's quite light and remains suspended in the media. And then we have these dense compact flocks that are in the optimal range. Okay, but where this sort of experience and intuition of the operators can break down, as demonstrated by the row below, is that we can also have images of samples from the activated sludge that fall within the same category, but look rather different to other images in the same category. So we can see that this looks rather different to that, and this looks rather different to that, but they are both fall under the same SVI bracket. So what we propose to do is trying to remove the subjectivity via human inspection. So can we quantify and automate the assessment of sludge health? And can we identify the emergence of filaments in unhealthy sludge and therefore remove the subjectivity of this assessment? And so with my colleagues, in particular Ryan Morris at the University of Edinburgh and Paul at Veolia, we have won a proof of concept award to try to use machine learning algorithms to help us do this. And we call this the Sludge Characterization platform. So why would we use machine learning in wastewater treatment? So machine learning is it's a statistics-based computational technique, and it learns from, so the algorithms learn from input data, and it needs large libraries of input data to do this. And as a result of then training the models, you can produce, or training the algorithms, you can produce models that aim to classify data that it hasn't seen before. So the archetypal example would be cats and dogs. So we can say here, as humans, we know that this is a cat and this is a dog, even though they share very similar properties. They both have the ears, eyes, nose, fur, etc. All right, and we can have many different images of cats and dogs. All right, they all the cats look similar to each other, but they're rather different, and likewise with the dog. And so these have been used, these types of problems have been used to develop machine learning over the last 20 years or so. And it turns out that you can put images into a computer algorithm of cats and dogs, and the algorithm will be able to differentiate the image as a cat or a dog. Now, if you can do that with cats and dogs, then you can obviously do this with healthy and unhealthy images of flocks in wastewater treatment. And that's precisely what I'm going to show you now. So this is work that we did with a project student. So we need a large library of data. And so Veolia operate many sites in Scotland. And in 3 of those sites in 2020, they sampled their sludge almost daily and and visualized under the microscope. Okay, so at this site, Alan Fairn, we have almost 800 images, and corresponding to each of the images, they collected daily data on the SVI. All right, so there is now data corresponding to each image, whether it is low, good, or high. And that therefore allows us to classify each of our images as high, good, or low. All right, so this is our library of images that we use as input into our machine learning algorithms. And it turns out that the low classification has a rather low number of images. So we don't consider that because of sampling, error in sampling statistics. And this gives rise to a binary classification system. So we will use these two sets of images, good and high, or cat and dog, to train a machine learning algorithm that is able to predict based on an image whether it is good sludge or unhealthy sludge. So the machine learning algorithm that we use is a very commonly used type of machine learning algorithm called convolutional neural networks. So it is a supervised learning technique to train on labeled data. Now our data is labeled. We have images that are labeled either good or high, all right? And it developed from this, we use typically between 10 to the 2 and 10 to 10 parameters or weights to fit the model. So how it works, so here is a schematic. Now ignore the details here, it's very complicated, of a typical convolutional neural network. So what happens is that the image here, in this case, in number 2, is passed through the network. And on passing through the network, the algorithms extract features from the image, and then it goes through to the end here into this artificial neural network part, and where these connected layers classify the data. All right. So an image of the number 2 goes in, and at the end we say, what is the probability that this image was a number 2? And so initially, when you're training the algorithm, it will not be very good at doing this. And so there is an error associated with every classification. And what it does then, what the network does to train the algorithm better, is that it works in a way as to minimize this error function. And so eventually you end up with an algorithm that is well trained and that can hopefully predict and classify any images that go through. So in our case, we have healthy sludge and a large library of healthy and unhealthy images. And so we are able to then train our convolutional neural network by passing these images back and forth through the network until it improves its classification ability basically by minimizing this error function. And every time it goes back and forth through the network, we call that an epoch. Okay. And having trained the algorithm, okay, with these labeled data set, we then say, so I will give you an image now that is not labeled here of a sludge flock, classify that for me. And the algorithm can do that with some accuracy. So we used a Python interface for machine learning called Keras. And Keras is optimized for image analysis, and it's very user-friendly. And as I said before, myself and my colleague Ryan Morris, although we knew a little bit about machine learning, we certainly weren't experts. So we were very lucky to get this excellent project student here in the School of Physics, Diego Fernandez, who was a bit of a wizard. And it turned out to be the case that the student was teaching the supervisors. So he managed to develop a model with almost 300,000 trainable parameters across 6 layers of the network. And so what he did was he split our library of images into a training set, a validation set, and a test set. So the training set is 80% of the images. Then you have a validation set, which is 10%, and then a test set, which is also 10% of our images. And once you train the network, you then use the model that you trained to test on images that it hasn't seen before. And basically, the take-home message here is that if we look at the accuracy of our model, here plotted as a function of the number of epochs that we use to train the model, we can see that in 3 runs, our accuracy is always above 60%. So if we look at the mean here, all right, we can see that we can get an average of 73% accuracy after 15 epochs, okay? So if we train the algorithm with 15 epochs, and then we test the algorithm on, say, 100 images, it will predict, or it did predict, 72 of those images correctly. All right, and it had never seen those images before. If we look in one of the runs, we see that after 15 epochs, it in fact was able to predict with an accuracy of 82%. So that means with 100 images in the test data, 100 images in the test set, the model was able to say that it was able to predict correctly 82 of those images out of the 100. And so if we focus then lastly on this data point, we can look at what's called a confusion matrix. All right. So we consider 36 images in the test set. So here on the left, on the y-axis, we have the true label, and on the x-axis, we have the predicted label. So when the images were labeled good, all right, our model got it wrong 7 times, but got it correct 29 times. And when the true label was high, so that's these fluffy samples, our model got it correct, or the model that Diego trained got it correct 30 times, but only got it wrong 6 out of the 36. So in conclusion, we've developed a prototype sludge characterization platform to predict sludge health from wastewater treatment images. And so we've demonstrated the proof of concept with a 73% average accuracy. And in one case, we actually got an accuracy of 82%, which is very encouraging, though this is only a proof of concept. So moving on, short term, we would like to, we were limited by the number of images. So Paul has kindly reached out to some of his collaborators or Viola colleagues in Austria and the Middle East, and they are now providing us with more images. So the more images that we can use to train our algorithm, the better it becomes. And we may consider other machine learning platforms such as TensorFlow and PyTorch. Longer term, can we develop the sludge characterization platform to predict operational failures? So for example, can we use it to predict that the onset of filamentous sludge bulking, but there is a likelihood that it may occur? Can we make it dynamic? At the minute, we've only been looking at static images, but can we access dynamic information like bacterial and protozoan motility? And can we couple it also to other operational data sets collected at the plant, for example, like Paul demonstrated with these probes looking at salinity or dissolved oxygen concentration and microbial activity? And with that, thank you very much.
Brilliant. Thank you for that, Gavin. That was a really interesting talk. So we've got a couple of questions come in. I'll just do the one for the sake of time because we're quite close to our end date now. I'm very aware, Stanislaus, that's twice now that you've been missed out on the live question, so very sorry about that. So this one comes in from Stefan, who asks, another important parameter to be viewed microscopically is the presence of certain protozoa. So he says that operators use their presence or absence, as well as the abundance of other sentinel microbes, to assess how healthy an activated sludge is. Yes. Could you incorporate such information into your model, such as using fish probes of bacteria, but he's not sure about what you could use to detect protozoa? We have considered it, and something we may look at, but probably the thing that limits us there is that, so in all the images that we get from the wastewater treatment plant, there will be very few protozoa in them, and therefore we're limited by the stats. Right. No, because we need more images of protozoa in the wastewater treatment in the FLOCS and sludge sample to train the algorithms. So if there's only the odd protozoa, then it may add little to the development of the algorithm. Right, I see. But what we maybe not concerned with machine learning, but we haven't had conversations with Paul. We have expertise here in Edinburgh on assessing microbial motility, but particularly bacteria, but very rarely do people talk about protozoan motility. So we are thinking about ways to characterize that and use that as an indicator for sludge health.